Good morning, everybody. Okay, we are going to go ahead and get started here. So, just a quick reminder of where we left off on Tuesday. So we started talking about molecular transport across membranes. We covered simple diffusion. So that's when molecules don't need any extra help from membrane proteins because they are typically very nonpolar, very small molecules that can just easily pass through that bilayer. Then we started with passive transport, facilitated diffusion specifically. And we talked about how sometimes molecules need help crossing that layer. And so tiny little ions, polar molecules, need these things called channel proteins. Usually these channel proteins are gated. They're either gated by special ligands, so it requires a molecule to bind, and that's what induces a conformational change, and that channel opens, and that polar ion or that polar small molecule can enter the tunnel, and that tunnel is coated in polar properties. Or it can be gated by voltage. So this is going to be more common in neurons. So before we go on to carrier proteins, which they act the same, but they help transport larger organic molecules. So let's look at an example of a ligand-gated ion channel protein. Okay, so here's the outside of the cell. And these are the little ions or small polar molecules that the cell hopes to internalize. Here is your closed ligand-gated ion channel protein. You can see it's closed. This is, the elevator doors have closed. And so what ends up happening, the cell senses that the molecules on the outside are much more concentrated than the inside. So there is a really severe concentration gradient such that the concentration outside is much greater than the concentration of this particular solute inside. Now, this is just an example of when the outside concentration is greater. Molecules can definitely leave the cell under the same idea, just except the concentration gradient favors the inside of the cell. Okay, so this is an example of concentration gradient favoring the outside of the cell. So the cell is hoping to reach equilibrium, so it needs to bring some of those molecules in. And so what happens here is that this cannot let any ions or whatever this little polar substance in until the gate is open, until the pore is open. And here is the little bodyguard, the ligand that binds to the binding site of that channel protein. Once that ligand binds this binding site, it induces a conformational change, thus opening the pore and allowing the polar molecules to enter the cell. Okay, so what are these key terms? When you're describing ligand-gated channel proteins, you're going to talk about the ligand's binding. They bind the binding site. Okay. The pore opens. Let's say conformational change is induced. This causes the pore to open. That center of the pore is polar. And now that is what allows polar molecules to enter or to travel through to be transported. Okay. And once the cell senses equilibrium, so this is going to happen until equilibrium is reached. And once the cell senses that, the ligand dissociates. And we go back to the beginning, the pore closes. Okay. Okay, so there's your example of a ligand-gated ion channel protein. So these would be little ions. Okay, that's also an example of a uniporter, right? Because it's one molecule entering one way. It's that protein only transports one molecule. So let's go ahead and talk about these carrier proteins. Same idea, but slightly different. They transport larger molecules. So in carrier proteins, give me 1 second. Okay, the pore is also, so these are organic polar molecules. The pore is also polar, because if it wasn't, then those nonpolar molecules don't need a carrier protein. Okay, so some examples of the types of molecules that can be transported are sugars, usually monosaccharides. And you're going to say, well, those are pretty small. Yeah, true, but water is 3 atoms. A monosaccharide, for example, glucose, is C6H12O6. That's 24 atoms, much bigger, even though it's just one monosaccharide. So a monosaccharide, individual amino acids or small peptides. Okay, and so those typically get transported by neurons, okay, because they serve as neurotransmitters. And the sugars, they need to enter our cell because the cell needs to break those down and make cell energy from them. And so let's see, the rate. So how is the rate affected? And so the rate really is affected in the same manner as channel proteins, right? So in the beginning, you're going to have this really nice increase as you increase the substrate concentration, but then you run out of available carrier proteins, and the carrier proteins are like, you gotta wait in line. So the rate flattens out. Okay. So again, the rate depends on the availability. So I'm gonna say same. The availability of carrier proteins, right? Because the carrier proteins have to be open, but if they are busy transporting things, they can't transport more stuff. Okay. So in this case, nothing is gating them. Nothing is keeping them closed. In this case, they don't need an extra molecule or an extra voltage change. Usually it's the molecule itself. So in this case, the cell senses a difference in concentration gradient, okay, whether it's high on the outside, low on the inside, or vice versa. And when it senses that, the molecule itself, that the concentration is off, for example, glucose. So if the concentration of glucose is off, glucose itself will bind to the carrier protein, inducing the conformational change. It opens the pore and the glucose can enter. So we'll just say the molecule that gets transported is the molecule that activates / opens the carrier protein. Okay, so like I said, the example that we can think about is glucose. So glucose does use carrier proteins, and so glucose does get transported via these carrier proteins called GLUT GLUT carriers. And so it's a family of proteins, and there's 4 of them. So it's GLUT1, GLUT2, GLUT3, and GLUT4. And so different GLUT carrier proteins are on the surfaces of different types of cells. Why, I don't know. But here's what I do know is that GLUT1 is expressed very commonly on the surface of human cells, and we can even trace it back to evolutionarily older organisms. So we say that it is highly conserved over time, or it has been highly conserved. And really, this is how, when we need to store sugar as glycogen, so we store sugar as glycogen, right? Stored glycogen. This is our energy storage. We store it in the liver. Well, how does the sugar get to the liver? Because you eat the sugar in the form of food, then your enzymes break it down into individual monomers, and then that glucose travels through your red blood cells, through your red bloodstream. So it needs to go from the red blood cell into the liver. So if it's traveling through the red blood cell, it's highly concentrated in the red blood cells, and inside the liver, if the liver's like, I need more glucose, somebody. So that means the concentration of glucose is low in the liver, and so that's how the glucose travels into the liver, through these GLUT1 carrier proteins. It can't just simply poof, pop into the liver. Okay, and so the individual molecules of glucose enter the liver, and then the liver puts it together as glycogen, because glycogen is a bunch of glucoses chained up by these glycosidic linkages. Okay, so this would also be an example of a uniporter if we're talking about one molecule being transported by one protein, and that molecule only goes one way. Okay, so this would be a uniporter. Again, just kind of emphasizing the idea that you can classify molecules a bunch of different ways. You can call it a carrier protein, you can call it a uniporter. So the carrier protein would more so describe the type of membrane protein it is, and then the uniporter would describe the type of transporter protein it is. I know, it's not very straightforward. Well, it is straightforward, it's just there's lots of options. Okay, so let's take a peek at this carrier protein in cartoon style. So what we have is, here's your molecule of interest. Like I said, the example that we're going to use in class is glucose. So maybe this is the glucose that the red blood cells just released. And here's your liver cell. And the liver's like, I need to make more glycogen, I need glucose. So the liver cell's sensing that there's lots of glucose on the outside. It's like, well, I don't want the blood sugar too high, so let's bring some of that glucose in here. And so here is your carrier protein. We always start at the beginning, and you can see, you might say like, it looks open already. Kinda, but it's only open on one side. So this glucose molecule can enter the open side, and once it does, so here, travel through time, now it's inside the pore. So this is polar, right? So sugar molecules have a bunch of hydroxyl groups. So we know that this is going to be a polar molecule. And then just to remind ourselves that the surface here, if you want glucose to enter and be attracted to that region, this also has to be polar. Okay, polar properties. And the converse of that is that if this is going to exist in the bilayer, if it's going to exist within fatty acids, this part is going to be nonpolar. Okay. So glucose enters the polar pore, and what does it do? It induces so glucose enters, or the molecule enters the polar pore. This causes a conformational change. So the rest of the protein opens. When the rest of the protein opens, the molecule can finish being transported. Okay. This keeps happening until equilibrium has been reached. Okay, so equilibrium. Equilibrium, you can see here as the molecule exits the pore, the top part closes. So as the cell reaches equilibrium, another conformational change occurs such that the pore closes. Okay. And then if the concentration gets too high again, then the whole process repeats. Okay. All right. Again, just to remind you, this is passive transport. So passive transport means, don't forget, whether it's simple or facilitated, that net cell energy is not required. Okay. And molecules are traveling with their concentration gradient from high to low. All right, that's passive transport. Cool. So since we're finishing up this lecture, I'll just take the rest of the lecture questions at the end, if that's okay with everybody. All right, so active transport. This is obviously different. If I just said, if it's passive transport, that means cell energy is not required. Well, if it's active transport, cell energy, net cell energy is required. Okay. And so that cell energy can come from a variety of different sources. And so you'll say like, wait, isn't ATP cell energy? Yeah, but that's not our only cell energy. That's just the one that everyone knows. But after this class, you'll know that there's other types of cell energy. So we'll say ATP is the most common for now. But know that there's other types of energy that can be used too. Okay. So cell energy is net cell energy is required. is required. Why? Because the cell is actively pushing molecules against their gradient. So the molecules being transported go against their gradient or up the concentration gradient, which means from low concentration to high concentration. Like, why the heck does it want to do that? Well, the cell has a lot of reasons for doing that. It's usually so that organs can function. So let's get into a little bit of some more details about this. So that's kind of the entire mechanism. That's the main difference between, the biggest difference between passive and active transport. But you can also think about it in terms of the types of transport proteins. Okay, so where passive transport, just as a real quick reminder, typically uses uniporters, active transport uses symporters and antiporters. So symporters are also sometimes in some textbooks called co-transporters, and they also use antiporters. And just a quick reminder what that means. Is that a symporter is going to transport two different molecules in the same direction. Not necessarily out to in, just in the same direction. Antiporters is a carrier protein that's in the surface that transports molecules, two different molecules, but in opposite directions. Okay. And I'll say mostly two different substances, but there's some fancy proteins in the surface of our cells that can do three. Crazy. Okay. So we'll go back to active transport. Active transport typically uses symporters and symporters/co-transporters and antiporters. And this is a two-step process. Okay, so it's 2 steps, meaning it requires 2 transportations. Transportation 1 sets up the cell for transportation 2. You cannot explain, you cannot have the transport of the second molecule without the transportation of the first transport protein. Okay, and we'll see what that means in just a second. Okay, so you have one protein, so this is usually one membrane protein, and it has a specific role. That role, it changes kind of the environment of the cell. Okay? So we'll just say change cell environment. The way that it changes the cell environment is what activates, so this right here is what activates the next transport protein to do its job. Okay, so transport molecule number 2 can work. Okay. All right, so let's get into a little bit more of an actual example. And maybe you've heard of this before, but the sodium-potassium pump. I'm certain if you've taken animal biology, you have heard of the sodium-potassium pump. And if you haven't, I'm gonna have a little chat with Blake. Okay, so here is what we're describing. We are going to talk about how glucose gets actively transported. So in the other example of passive transport, I explained how glucose gets passively transported. So one thing already to keep in mind is that in specific places and times in our bodies, in animal bodies, glucose can use passive transport. And sometimes we need, so this is from high to low concentration, and sometimes our cells require active transport of glucose. Okay, which means we use some of our cell energy because we're forcing glucose to go from an area of low concentration to high. So to transport glucose in an active mechanism, you can't just do it straight up. You need something to happen first, and that is going to be what we call the primary transport of glucose. And if you can see here, we're talking about this thing called a sodium potassium ATPase, otherwise known as the sodium potassium pump. Anywhere in there do you see the word glucose? No. So this first protein that's involved has nothing to do directly with glucose. It does not transport glucose at all. Okay, so here's another lesson in how we can categorize proteins a bajillion different ways. So it is a carrier protein because it's in, well, I guess it's a channel protein because it's transporting ions, right? So this is an example of a channel protein because it's transporting ions, but it's not gated, right? I'm going to tell you that right now it's not gated by any certain thing. So it's a different kind of channel protein. Next, you can see from the picture here, let's say you can see here we go. Sodium is going one direction and potassium is going another direction. So not only can you classify this as a channel protein, but more specifically, you would classify it as an anti-porter. Remember, I just said that active transport usually uses sym-porters and anti-porters. Okay, not only that, but it's also an enzyme. You can tell that it's an enzyme because it ends in ASE. And so what kind of enzyme is it? It's an ATPase, which means that it cleaves or hydrolyzes ATP. So this means that somewhere in this whole reaction, you're hydrolyzing ATP into ADP plus an inorganic phosphate. The reason that this happens is because when you cleave that phosphoanhydride bond, you release energy. And what did I say was one of the biggest differences between active and passive transport? The use of cell energy. Where did I say it typically comes from? ATP. Okay, so this thing is called, the name of this pump is called the the sodium potassium pump. And then why do we call it that? Because it's pumping those things. All right, so let's see how this thing works. And so I really, really like this cartoon, and I know it kind of looks busy to start with, but we'll just go through it step by step. So the first thing to really recognize is the concentration gradients of each of these things. So I'm going to highlight them in different colors. So let's say that we're going to highlight the sodium in green. Okay, so for starters, the concentration of sodium ions is higher outside than it is inside. Okay, so it's high outside, low inside. And then for potassium, it's the opposite. It's low outside and high inside. Okay. So maybe I think this might help you remember it. So let's just highlight where the ion concentration is highest. Okay, so there we have it. High sodium outside, high potassium inside. And then we'll take this step by step. So how does this pump work? So here's the pump, and a couple other pieces of, I guess, terminology that you should use is, remember, this is extracellular. This is the outside of the cell, and then the inside of the cell we call intracellular. Okay, those phrases are the way that you formally communicate the science. And so what happens here is the cell senses that it needs to transport glucose. And so what it's gonna do is like, okay, well, we gotta set it up. So how do we do this? And this time the cell's actually not going to be trying to reach equilibrium. It's crazy. It's gonna actually try to make this higher on the outside and then potassium higher on the inside. It's trying to make the concentration gradient even more severe, which is nuts, but you'll see why that's important. So this is all in an effort because the next step is transporting glucose. So here's your pump, and exactly three sodium molecules, 3 sodium ions bind to the intracellular region of the pump. So here they are. At the same time, or very close in step, you also have an ATP that binds. Okay. So if I had to draw this out, maybe it would have your phosphate, 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 and then the rest of the molecule. Okay, that's ATP, right? 3 phosphates, the rest of the molecule. Okay, and so what ends up happening, remember that this thing is an ATPase, and so it cleaves ATP. And so what it's going to do is, let's zoom back in, and it cleaves that phosphoanhydride bond, boom, 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 uses water to cleave that, hydrolyze it. And so that's what's kind of been showing here is the phosphate group stays there. So here's the little phosphate group that stays there. And the ADP just goes into the cytosol. But remember, you don't just have phosphate, inorganic phosphate, you also release energy when you did that. So the enzyme's like, cool, now I have energy to do my job. This is awesome. And so the important thing about the phosphate group is that once this pump gets phosphorylated, this actually makes the conformational change occur. And I think that's what's written here. So yes, the hydrolysis of ATP changes its shape. So that is synonymous with saying induces a conformational change of the pump, such that now the sodium molecules can enter. So they were on the outside, it cleaved the phosphate off. Now it's phosphorylated, now it's shape changed in cleaving the phosphate off. Now it has energy, cool. So these molecules enter the pore. Again, it's a polar pore. And so in doing that, this actually, when the molecules enter here, they are going to be released. So here they are being released into the extracellular region, and releasing that now induces another conformational change. So these guys right here being released induce another conformational change. This conformational change allows potassium ions to bind, but there's 2 of them. They bind on the extracellular portion of the pump, right? So these bind the intracellular portion. And so they bind at the extracellular portion. And again, the same thing kind of happens. Once they bind there, okay, so they bind here, and then it's not shown here, but a slight conformational change, more conformational changes occur such that now the potassium ions can enter the pore. And once they enter the pore, this is this is what allows them to leave and enter the intracellular portion. So releasing potassium to the cell's interior now is going to actually assist in dissociating the phosphate group. So this will dissociate from the pump, and so this, again, these two steps kind of happen at the same time. So the potassium molecules kind of enter. As they're entering, the inorganic phosphate is detaching or dissociating. This causes a slight conformational change so that the potassium ions can leave. So let's see. We'll say that the potassium ions in the pore cause the phosphate to dissociate. At the same-ish time, this causes another conformational change so that the potassium ions can leave or exit into the cell. Okay. And then once all that happens, I'd like for you to appreciate that once this leaves, look at the shape of this. It looks like this. Imagine those little sodium ions weren't there. So it returns the pump to the same shape that can now again take 3 more sodium ions, attach an ATP, and repeat the process. So this really goes full circle. Okay. So just to kind of clarify, these are not 5 different pumps in a row on a cell. It's showing you as these are like snapshots through time. Okay. But it is important to recognize that we don't just have one sodium potassium pump on our membrane. We have hundreds, if not thousands, because this is happening all the time. Okay. And so again, the sodium potassium pump is found in animal cells and is really important for we say that it maintains proper concentration gradient of sodium and potassium. Okay, so great, we just explained it, but what was the overall outcome? Okay, so just briefly recap, you took sodium, which was already low in the inside, and you scooted it outside. So at the very end of this, the concentration of we did sodium in green. Green. At the very end of this, the concentration of sodium is lower on the inside and even higher on the outside, right? Because you sent 3 molecules outside, right? And then the opposite is true for your potassium. It's even lower on the outside and higher on the inside. Why? Because you sent 2 molecules in. So the cell's nowhere near equilibrium. It's now even further from equilibrium. So what the heck? Why did this have to happen? And so the take-home message here is that the sodium-potassium pump functions to establish a very severe or significant or grave concentration gradient of sodium such that it is very high outside or in the extracellular region versus the intracellular region. That is the goal of the entire sodium-potassium pump. And it's this. This is what activates the next transporter protein. Okay, so remember, this is all for glucose, all to transport glucose. So the other thing I want to just point out is in both cases, these ions, both ions, so both sodium and potassium ions were moved against their gradient. Right? They both went from low to high regions, both of them. Okay. Now let's see how that contrasts with the sodium it's called the sodium glucose symporter. This already gives you ideas of what's going to happen. So this tells you sodium's going to be transported, glucose is going to be transported. If it's called a symporter, then they're probably going to be traveling in the same direction through the same protein. And that's exactly what happened. So remember, in the previous step, the whole entire goal was to make the concentration of sodium really, really high outside versus inside. And so here we can see all of our sodium, so much sodium out here, and just a few little speckles inside. Okay, so the cell now the cell wakes up and thinks like, oh my God, we're nowhere near equilibrium. What the heck? We gotta fix this. And so this pump here is what is activated. That's what senses this severe concentration gradient. It's a completely different, sorry, this one right here. It's a completely different molecule. This is the sodium-potassium pump right here. Okay. This one is a completely different molecule that's usually right next to sodium-potassium pumps in our membrane. It's like, oh my God, sodium-potassium pump, well, you just like took us so far from equilibrium, I have to fix this. Thanks for nothing. Thanks for extra work for me. So essentially what ends up happening is this senses the severe concentration of sodium on the outside, and this is going to transport sodium. This time, sodium is moving with its gradient, right? Because it's going from high to low. That's what this pump does. It's going to transport it from high into the cell to try to reach equilibrium. In doing that, so you're gonna say like, well, this is active transfer, so where is the energy coming from? That concentration gradient is the driving force. It is the energy that kind of forces the pump to work And as the sodium's coming into the pump, it's dragging with it glucose. And so glucose, we are actually transporting glucose from a region of low concentration to high concentration. So in the secondary transport, sodium, we say sodium is traveling with the gradient from the exterior, the extracellular region to the intracellular region. So if it's going with the gradient, it's going from high to low. And then this is so that this is the energy, the gradient is the driving force that causes or allows the pump to travel, to transport glucose against its gradient. And that means from a concentration of low to high. And that is also going from the exterior to the interior region of the cell. So both of them are going from out to in, which is what makes this a symporter. But importantly, you can see here that one molecule is going with its gradient, and that is the driving force. This was established by the primary transport pump, and glucose is going against it. So hopefully now you can understand or see or believe me that primary transport had to have happened, because if you didn't have primary transport, the concentration gradient of sodium wouldn't be enough. And so if it wouldn't be enough, it would never activate this pump and never allow glucose to enter the cell. Okay. And I can't play these videos during class because we are recorded, but there are some videos for you, and this kind of shows you the dynamic process. So I show you screenshots of this happening over time. So this is kind of showing you how it happens in our cells. Two different videos, and just to let you know that this video does call it a co-transporter. I don't know. Okay. All right. I think we can skip this. But it's really nice. So I would encourage you to look at it to see how this kind of relates to us as a human being, right? Or as animals that consume food and digest it. And we have things that we have to worry about like blood sugar. So if you just go through the cartoon here, it's really just trying to explain to you how when you eat food, the food goes to your belly, and then inside your stomach, you have these enzymes that break it down into smaller components. And then those smaller components get absorbed into the stomach and the small intestine. But if we're trying to get blood into the bloodstream and into the liver, this kind of explains how that happens. And I think it's really, really cool. It's a really cool cartoon that I encourage you to look at. Okay, so I'll take some questions about primary. Wow, we were really behind. Primary and secondary transport as well, whatever we've talked about today. So are there any questions? Okay, so is active transport smaller or larger molecules? So we started really, really tiny with passive simple diffusion, and we're just getting bigger and bigger. But active transport, you know, you just saw that active transport just transported ions, which are tiny, and it transported glucose, which is a little bit bigger. So I can't really answer that question. It's both. Okay, Lucy.
Why does the secondary pump why does sodium pull glucose with it?
That's the whole purpose of the concentration gradient. So you would also see this happen in neurons. So neurons, remember we said that there's like this this electrical potential on the inside of the cell versus the outside of the cell, and when that gets too brave, pumps open, and then the ions can change, so it can kind of reach equilibrium, but that's also what allows pumps open so that neurotransmitters can leave. It's just gradients are just driving energy forces.
Right, I understand why it would pull sodium back in, but I don't think I understand why glucose would go with it.
Because this pump is specific to sodium and glucose.
Okay, okay. All right,
so the sodium potassium pump sets up the gradient for sodium glucose transport. That's it.
Okay, thank you.
Okay, Max.
Does potassium have any role in the secondary part of the?
No, not directly. Its role is in primary transport. So we say potassium has an indirect role because it had to have been a part of primary transport.
Thank you.
Annika, is the ligand-gated ion channel a uniporter only? It depends. So it just depends on how many molecules it's transporting. So if it's a ligand, in this example that I showed you guys today, this is a uniporter. But are there transport proteins that require ligands to open and then also send two things either way or both ways? Probably. So again, don't try to fit it into a single box. Okay. Any other questions? Cool. Okay, so those were like small, medium-ish molecules being transported. Now we do, here's our summary of that, right? So we have tiny non-polar or the super VIP water using diffusion. Other polar but small molecules, either using channel proteins or carrier proteins. Again, this cartoon, I hate that they only show things going from outside to inside. Please just remember that molecules are traveling in both directions and there's pumps that pump stuff out of the cell too. Okay, active transport, the summary here is just showing that multiple molecules are involved, energy is involved, and this doesn't really show, but remember this is going to use cell energy because things are being pushed, at least some of the things are being pushed against their gradient. Here, everything is being pushed with its gradient, down its gradient, from high to low, so we don't use net cell energy. But we, as cells, we have to transport larger things too, whether that's getting it out of the cell or getting it into the cell. This is, if we were just transporting 2 or 3 molecules at a time, man, we'd be sitting here for forever. So we're just going to briefly recap some of these mechanisms. And so because they are, I guess, bigger processes, in general, endo and exocytosis are energy requiring processes. So endo and exocytosis, both of these use cell energy. And this could be by means of ATP hydrolysis. It could be means of other types of cell energy. So just know that it uses net cell energy. So let's take a peek at this. Just the overview of endocytosis is endo means indoors, enter. Okay, so it's bringing stuff inside. And so here this cell is trying to eat this delicious meatball. And so it's too big to use a transporter protein. So this is giant meatball, okay? Too big for a carrier protein, too big for any kind of transport proteins. It's too big. So what does the cell do instead? This is, again, when it drives the importance of the fluidity of the membrane. So if the membrane was this rigid, stiff thing that didn't have any flexibility, none of this would be happening. So essentially what you're doing is the membrane's kind of like hugging the meatball. Come to mama. So it hugs the meatball, and as it's doing that, it's forming this nice little protective vesicle. And so this vesicle is what allows big fat molecules to enter the cell. Okay. And so there's different types of endocytosis. So one that we're going to go over in class. So one very common type of endocytosis is called phagocytosis. And these are going to be mainly used by cells called phagocytes. And so you may already know this, that phagocytes really are a way to kind of ingest or engulf exterior material and get rid of it. So here is an example of that happening where you have, this is the exterior of a white blood cell. Maybe I could draw it nice and big here. Here's your white blood cell. And it has recognized a foreign pathogen like MRSA. So these antibiotic resistant bacterial strains. And so the white blood cell knows it's time to get rid of that. And so essentially that's what it's trying to do, is it's trying to kind of wrap that around and it's going to get rid of this. Okay. All right, so another example. So this is to get rid of pathogens, right? So this is immunity, get rid of pathogens, right? So this helps us stay alive. Okay, and then we also have other examples where organisms can undergo phagocytosis so that they can receive nutrients. So you have your amoeba, which is a protist, which is a single cell eukaryote. That amoeba has to eat. It doesn't have a mouth, it doesn't have a belly, so it needs to get nutrients from somewhere. So the process by which it gets nutrients is phagocytosis. And so here is the amoeba, his little fake lip trying to curl around whatever that thing is. I don't know, okay? Ball of cheese. All righty. And so these phagocytosis is typically going to be big, large molecules. Okay. All right, so we'll say large or whole cells, right? Because MRSA, this is a whole cell, but the white blood cell is much bigger. Alrighty, so pinocytosis. Pinocytosis is, here's a nice little picture here. I think it's Greek. I think it means liquid. I don't know. Don't quote me on that, even though it's on YouTube and you can't quote me. So how is it different from phagocytosis? It is different because the molecules themselves are not bigger, but the goal here is to ingest or bring in more than just a couple molecules at a time. So it's basically going to be some kind of liquid with multiple molecules, not multiple, maybe sometimes different molecules, but basically like not just one glycine neurotransmitter, but like 100 copies of glycine neurotransmitter so that we can get lots, like do it fast. Give me a lot right at one time so I don't have to sit here and wait. And so this is the type of endocytosis that is going to be used by how blood kind of transports nutrients to different parts of our body. So this is how like intestinal cells uptake, absorb, whatever, nutrient molecules from blood cells. Okay, so that is shown right here where you have the inside of the cell, but the same idea happens. So here are your smaller molecules. They're traveling through liquid like blood or lymphatic fluid or whatever it may be. And these molecules, again, the membrane kind of surrounds. Drink, Pino, Greek, drink, hungry. Okay, see, I was kind of there. So the membrane kind of pinches around these molecules here, forming a vesicle. Each time you can see that a vesicle is formed, the vesicle is made of a phospholipid bilayer. Okay. And here's an actual real picture of this happening where the vesicles are forming right here. So here's the vesicle forming around. You can't see the individual molecules in the liquid, but here is the vesicle forming. Here's the vesicle forming. Okay. Alrighty, and then the last kind of mechanism that we're going to talk about for endocytosis is something called receptor mediated. So we know that vesicles are definitely going to be involved, but this one is a little more complex. And the receptor-mediated endocytosis is going to be very common in neurons. So when we were talking about the Kikuchi et al. Paper, and we talked about those two different neurons, how one neuron releases something and then the next neuron is supposed to grab onto that dopamine and bring it into the cell. Well, it's not just one dopamine at a time. And so you got to remember that many things are going to be, so if the top neuron is here and the bottom neuron is here, this space in the middle is called the synaptic cleft. There's going to be a lot of molecules floating around in that synaptic cleft. And so if a neuron has a specific neurotransmitter that it's only supposed to receive, it needs a way to only receive that neurotransmitter. And so what we have, so here's your cell membrane. And this is not just in neurons, but it's the most common example. So here's your cell membrane. And you can already see the pinching off. So you know that a vesicle is going to form. But if we zoom in on this, what is special, and this is going to be present along the membrane, is you have these little receptors. They're like tiny little catcher mitts. Okay, and that catch like a baseball mitt, well, should only catch a baseball. It doesn't catch a football. It doesn't catch a golf ball. It could catch a golf ball, but shouldn't. Okay, so imagine it could only catch a baseball. And so the ligand here, the molecule that needs to be ingested or endocytosed, can only get in there if it attaches to the receptor. So once this interaction happens, that's when the cell membrane starts to form a vesicle. So I guess the order is the ligand binds to its specific receptor, and then this causes the pinching, which is basically the start of forming a vesicle. Okay, but let's see what's on the other side. So this is the extracellular region and this is the intracellular region. So there's some magical stuff over here. What is this? These things are called clathrin. So we have clathrin intracellular, right underneath the phospholipid bilayer. So clathrin, we say that the phospholipid bilayer on special cells are coated, right? It's coated with clathrin. So oftentimes you'll see a clathrin coated pit, as the phrase, people call it a clathrin coated pit. And so when this bubble kind of forms, so it's pinching, 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 basically it's gonna pinch until it makes a complete circle. And now this vesicle, instead of these being on the inside, because of the way they've hugged the neurotransmitter or the ligand, now these clathrin molecules are on the outside. So clathrin is a type of protein. Okay, these are all clathrin molecules. And then you have your ligand secure inside the bubble. And so this is going to then fuse with the endosome. And so when it fuses with the endosome, a couple things can happen. So you can kind of, it basically separates like, okay, ligand, you're nice and protected. You can go ahead and release from the receptor. And so you can send the molecules to wherever they need to go. And then the remaining receptor will kind of be recycled and into its own little vesicle. And then this vesicle refuses with the membrane and then the receptor's back on the membrane surface. So we say, so refuse with membrane. Okay, cool. And then the molecules themselves will go on to wherever they are needed. Okay. All right, so that's receptor-mediated endocytosis, most common in neurons, requires clathrin coats. And so the clathrin, what we think is that the clathrin is responsible somehow, I don't know how, is responsible for mediating where the cargo will go. Where the cargo will go. Okay. Great. I think that's it. Cool. Now, last but not least, we have our summary of endocytosis, where you can see phagocytosis are these big, large particles or whole cells, too big for protein receptors or protein transporters. They are going to be engulfed. And so oftentimes if they're food, we'll call them food vacuoles, but in the case of white blood cells, it's phagocytosis, it's an immune response. Pinocytosis, it takes in some of that extracellular fluid because you need more than one molecule at a time. So this is a dumb cartoon. Let's draw a bunch of these. Okay. Okay, and then we have a receptor-mediated endocytosis where you have very specific receptors. See, this green receptor only likes the stars. It's not going to receive the blue squares. And on the inside, you have the coated pits so that when you actually form the vesicle, the coated pits are now, the coated clathrin is on the outside, and you have your special star molecules on the inside, and they can each go their kind of separate ways. Okay. And I don't think we need to do a lot of talking here because it's basically the opposite. So exocytosis is just the way that a cell exits or makes things leave that don't fit through protein receptors. or transporter proteins, but the same idea. So you would form a vesicle from places like the endoplasmic reticula or the Golgi, or neurons do this too. When they're packaging up their neurotransmitters, they will package them up too. So here are your molecules of interest surrounded by this really nice phospholipid bilayer in a vesicle. And so this vesicle gets directed towards the cell membrane and fuses with the cell membrane. And then once it fuses, basically that vesicle's popping open and all the molecules can exit and go where they need to go. Usually if it's trash, they get thrown out. If it's a signaling molecule, these molecules will go to other cells to do whatever they need to do there. Okay, so questions on transport of large molecules. All right, sweet. So, okay, so we are starting lecture 9. And so lecture 9, we just finished talking about how molecules get transported across the membrane, whether they're coming into the cell or whether they're leaving the cell. And that kind of goes hand in hand with what does the cell do next, or why did that have to happen? Remember, my 2 favorite questions are why is that important and how does it happen? So we kind of just talked about the hows of how molecules transport. Now, why is that important? Well, if you're a multicellular organism, even if you're a single cell organism that lives in colonies, you have to have a way to communicate to cells that are nearby. If you're a multicellular organism, not only do you have to communicate from one cell region of cells, the brain, to the rest of other regions of cells, the rest of your body, but you have organs, right? And you have tissues and you have skin. You're not just a blob, you're not a goopy mess. You have organs that stick together in a shape. Your heart is in the shape of a heart. And so the way that cells make a heart is they have to stick to one another. So this is called cell adhesion. So sometimes your cells have to stick to one another to form tissues. Sometimes your cells have to stick to other environments so that they don't float away, so your heart doesn't go floating around. And definitely cells have to communicate. And so really these are 2 different things, but they're important. So I don't know if you can tell by now, but I'm a huge Laker fan. So this cartoon here kind of shows what we're going to talk about today. So you have cell 1 and cell 2. They are showing or demonstrating adhesion. And so this is a way that cells can form a little team or a community, right? And so here, if you're old like me, you will recognize this is Magic Johnson, and he is a Laker from the 80s. And he is communicating with another cell, but he's actually not touching that other cell. So sometimes cells do have to touch to talk, and sometimes the famous no-look pass, he's sending the signaling molecule to that cell without touching him. Okay? So those are pretty much the 2 things that we're going to talk about: cell adhesion and cell communication, right? Via longer distances. Okay. So here's the overview. And I think that this is really important because it's like, let's look at it in the beginning and then let's look at it at the end, because this is a lot. I'm going to be real honest with you. It's a lot. So 2 reasons why cells interact. They interact to stick together and form tissues, or they interact because they need to communicate so that cells can change behavior in response to some certain environmental changes. So there's 2 types of interaction. There's interaction with the cells are physically touching, physical contact. This is called direct cell contact. And there's different types of direct cell contact. And we're going to go through each of them. There's something called tight junctions, adherens junctions, gap junctions, and then finally these things called focal adhesions. I pair them with something called hemidesmosomes. So the adherens junctions are not synonymous. They're similar to these things called desmosomes. And focal adhesions, see adhere. Focal adhesions are similar, not the same, similar to things called hemidesmosomes. Okay, and so next to each of these structures, it kind of just outlines what each of them are important for. So these are all direct contacts, but some of them are strictly so that the cells stick together. Some of them are mostly for sticking together, but a little bit of talking happens. Other ones, yeah, the cells are physically contacted, but it's not like a really tight bond. So the cells, if you really wanted to, they could kind of separate from one another. So this kind of junction is mostly for communication. And then this special one, which is why I left a gap here, is for cells to communicate with their environment. So it's not cell to cell, it's cell to extracellular matrix, right? So cells don't go floating around in the body. All right, then the other type of contact is called indirect cell contact. So if I'm contacting you via email, I'm not actually holding your hand and talking to your face. I am indirectly communicating with you through the World Wide Webs, okay? The interwebs. So you have indirect cell contact. This is strictly going to be for signaling and communication. These terms are used interchangeably. So these kind of like, they mean the same thing. It's how cells signal to you. Like when I say like, I strongly encourage you to attend PALS, that's like me signaling to you like, you should probably just go. And then that's me also communicating that to you. All right, so all of these types of interaction, if it's indirect and the cells are not actually touching, they require ligands and receptors. So something is going to be binding to a protein. That's what starts the line of communication. So it's kind of like the subject line, and then you hit open, right? You have that the subject line is the ligand, and then your finger pushing open is the receptor. You're receiving my email. Okay, and so there's three mechanisms here. There's paracrine, autocrine, and endocrine, right? And they have different reasons, but they're all indirect, and they're all for communication, and they all use ligands and receptors. Okay, let's get started. So we'll start with the tightest of all the junctions, and then we'll loosen up until the cells are not even touching anymore. So I have this cartoon a couple different times or something very similar. So let me just kind of explain what you are watching or looking at. Okay, so here is a cell. It's an intestinal cell. So this is at the one surface of the intestine, you have these villi or villi. Okay, so you have your epithelial villi of the intestine. So this is an intestinal cell. This is an intestinal cell. Here's the little nucleus. Okay, so you know that your gut or your small intestine is not just like one solid tube, right? It's a bunch of cells right next to one another that form a tube. And in the center of that tube, it's hollow. It's called a lumen, right? It's not empty, but it is a space for things to travel through. Okay, all right. So the cells of the small intestine have to stay together. So here we're looking at tight junctions. And so for each of these tight junctions are typically made of protein structures. So that's in general. So let's just write that. Junctions, that is my abbreviation for junction, JX. They are going to be composed of proteins. Proteins with different names that you need to know. So for tight junctions, the tightest of junctions, so no communication at all, so no communication. This is strictly 100% for adhesion. Okay, and this is also we call it a 100% impermeable, not semi-permeable, That means not permeable. Impermeable. That's why no communication, no molecules, no signals, or nothing's happening. It's like taking a test. No one's talking to anybody, okay? All right, impermeable. So we say that it is a complete barrier. So no molecules are going this way. No molecules are going this way, nothing. So the types of proteins that are part that make up these junctions are called claudins and occludins. Okay, so that's, let's do it in red. Claudins and occludins. Okay. And as you can see here, the claudins and occludins span the entire phospholipid bilayer, because this part is the intracellular region and this part is the extracellular region. So by definition, this is a transmembrane protein. It's not responsible for transporting things. It's responsible for making a junction. So this is one claudins and occludins. Okay, so you don't have to worry about this for now, or for this class. So we'll just, we'll just put this in parentheses because I do want you to know that it exists, but we don't need to really know about it in this class. Okay, the next thing you have are actin molecules. So remember, actin is a type of structural protein. And structural protein, if you remember the word actin, is also part of the cytoskeleton, right? Cytoskeleton. So here's your F-actin. It's a special type of actin. It's called filamentous actin. So those are the things that make tight junctions. And that's pretty much it. So what you need to know about the tight junctions is that no, they're not for communication. And even if they wanted to be for communication, they can't, because the way that the claudins and occludins bind to the F-actin and the way that it really holds the two cells together, molecules can't go between cells, nor can it go from the lumen to other parts of the body, like the bloodstream. Okay, cool. So that's our tight junction. All right, the next type is our adherens junctions and our desmosomes. Right, and so that's shown here as well in this cartoon, and you can see some similarities. So I'll start with the function of the adherens junctions. And so one of the functions of the adherens junctions is, we say that it's, yeah, we can say that it's for adhesion. Adhesion. Okay. But this also plays a role in some communication. So if there's some communicating happening between these two cells, that means some molecules, probably tiny ones, some molecules can get through between one cell to the next. And no, adherens is not the same as gap. Some can get from one cell to the next, okay? So if a molecule can get from one cell to the next, then we say that it is semi-permeable barrier. Okay, and just to put it into perspective so that we all know that if you zoomed in here, this is where you'd have your bilayer. All the way around, so you'd have your bilayer here, all the way around, just to put it into perspective. How big carrier proteins are versus how big cells are, right? So a carrier protein is going to be this tiny little thing here. Put it all in perspective, etc. Right? So our cell membranes have a lot of junk going on. Okay, let's see. But in these adherens junctions, you have a couple different types. So adherens junctions, let's just take a peek what we have. We have these things called adapter proteins, and then these things called cadherens. And so this is just one type of adherens junction that they're showing. But here's the one similarity between adherens junctions and tight junctions is you still have attachment. So they're associated with the cytoskeleton. Right? So you have your, in this case, your F-actin. Right? Here's your F-actin right here. So it turns out, however, that adherens junctions, they use what are called, the proteins that they use for these junctions are called cell adhesion molecules. Really clever name, right? So this E-cadherin is a cell adhesion molecule. It's just E-cadherin is a type of cell adhesion molecule. Some of these cell adhesion molecules are dependent on the ion calcium, so we call them calcium dependent. Some of the adhesion molecules, thus some of the junctions, are independent of calcium, so we call them calcium independent cell adhesion molecules. Okay, so the ones that are dependent on calcium, we put them in a family of proteins called cadherins. So Ca for calcium, D for dependent, herins for I don't know, okay? So this E-cadherin, the fact that it's an cadherin tells you that it is dependent, so it needs calcium in order to bind to the E-cadherin from the other cell. That has to happen. It's almost like the calcium molecules are like the final little super glue that keep them together with the buttons, the snaps. Okay, so these cadherins, the other things that cadherins use, so cadherins, not only do they depend on calcium, but they also interact with a class of molecules called catenins. Okay. And usually these catenins, this would be your catenin. So a catenin is a type of adapter protein. And these catenins, what's their job? As you can see here, let's look. Here's your E-cadherin. So this yellow stick, here's your E-cadherin. There's a part that's intracellular, a part that's extracellular. So this is classified as a transmembrane protein because it crosses the entire phospholipid. It has the ability to attach to the neighbor cell's E-cadherin because let's draw little tiny calcium molecules there. Cool. Calcium. Okay. Here's your calcium molecules. Okay, but then I say they also interact with catenins. And so catenins, or your adapter proteins. And so these catenins, what do? You can see that the E-cadherin is attached to the catenin. And it's the catenin that's attached to the actin cytoskeleton. So you can see here, this does not physically attach to the actin. So the catenin is that's why we call it an adapter, because it's kind of like a bridge protein. It is what attaches, it kind of is like the middle ground, the person that, the mediator, right? It's holding the E-cadherin's hand and it's holding the F-actin's hand, and together they're one strong force. So the role of catenins, their specific role is to interact with the cytoskeleton. Okay. And so that keeps the two cells not only attached at their surface, but we know that the cytoskeleton is that strong skeleton, interior skeleton that kind of makes sure that the structure of the cell itself is nice and safe and sturdy. So the cells are interacting with one another and they're anchored to something really strong and rigid. Okay. And then the independent, I don't have room. The independent cell adhesion molecules, one example of these are your immunoglobulins, or your IG whatever. So if you've been following this whole COVID vaccine type situation, and they're talking about antibodies and your IgGs and your IgMs and blah, blah, blah. Those are immunoglobulins. Immunoglobulin is the fancy way of saying antibodies. Okay. So certain cell types make immunoglobulins and express them on their surface. And the way that you can, if you do get infected and you need to build an army of fighters against this infection, when you present the immunoglobulin on your surface, that immunoglobulin or that antibody can go and attack that pathogen. It binds to that pathogen and destroys it. But before you do that, one cell doing that is not going to do anything, because the viruses and bacterial strains, they replicate so often that there's billions, millions of them. So the immunoglobulins, there's lots of them on surfaces of certain cells, but the cells also need to replicate and divide. And so when you have cells that are trying to fight a certain virus, they usually, or pathogen, they usually will make the same immunoglobulins on their surface. And this allows them to interact with one another and go together to do their job. Okay. So we don't know if they interact with the cytoskeleton on the inside. That's kind of up for debate. There's some researchers that show data that that is the case, others that show data that that is not the case. So if we were drawing, let's say, here's your two cells, and then let's draw their little immunoglobulins that are on the outside. Cool. Immunoglobulins of the same type usually interact with one another, but we do not know, they go all the way through. We do not know if they interact with the cytoskeleton. Question mark. We don't know. Okay, but importantly, they are calcium independent. So it's not reliant, your immune system doesn't totally rely on calcium. Right? Okay, cool. So when you have these cell adhesion molecules, usually the same cell adhesion molecules are presented on the same cell type. So that means cells of the same cell type, like the epithelial cells of your intestine, they interact with one another, means they also make the same proteins on their surface for adhesion. So when we say that you have cell adhesion molecules interacting with one another that are the same, we call this homotypic binding. Okay, and here's your example right here where you have E-cadherin on one side interacting with E-cadherin on the other side. They're both E-cadherins. There's A-cadherin, there's N-cadherins. Those are not going to be usually made in on intestinal cell surfaces. So most times cells exhibit homotypic binding of their cell adhesion molecules. Certain cases, special cases, require different types of cell adhesion molecules to bind. If that is the case, so we'll say rarer, or we can see some different cell types exhibiting heterotypic binding. Okay. So if these are IgGs, that's going to be homotypic binding. In some cases, cells that make IgG on their surface can interact with cells that make IgM on their surface, and that would be heterotypic binding. If I said IgG, that's homotypic binding. I don't know if I misspoke. I feel like I might have. Okay, great. That covers the adherens junctions. And the desmosomes, you can see, are very, very, very similar. How are they different though? So if we look at the desmosomes, we still see those transmembrane type of proteins. Okay? So you still have those types of proteins here, and they still use adapter proteins. But the only difference really is the type of filaments they interact with, where adherens junctions specifically interact with F-actin. Desmosomes are going to interact with the intermediate filaments. Why? I don't know. Okay. But the rest is the same. They still have a transmembrane protein. that interacts with the neighboring cell transmembrane protein. They still use adapter proteins, and the adapter protein is what interacts with the intermediate filament. Okay. But they're still same, mostly for adhesion, mostly. And then there's some level, small level of communication, okay, which makes them a semi-permeable barrier. Not a semi-permeable membrane, a semi-permeable barrier that is in the membrane. Barrier. Oh, I have a better, okay, cool. Well, here's a better example of E-cadherin and really what's happening here. So here's cell 1, the inside of cell 1, the inside of cell 2. Okay, inside. Okay, and here's the little space between the two cells. And you have your E-cadherin. It's not showing what's happening on the inside of this cell, but here's your E-cadherin that spans across. And then you have, this is the adherens junction. Here's your little calcium molecule somewhere in here that's not shown, but it's there, they're there. And then you have all of the E-cadherin interacting with the catenins. CTN is catenin, so all these different catenins. You don't have to memorize alpha, beta, P120 catenin. Just know that cadherins need catenins. And then they need the catenins because it's the catenins that bind to the actin cytoskeleton. There's your adherens junction. And I'll just cover gap junctions and then we'll call it a day. So we will have concluded, oh no, let's finish. Gosh, curse words. Okay, yeah. I wanted to get through direct cell contact, but I think that would be pushing it. So let's just finish gap junctions. Okay, so the last type of direct cell contact that puts cells in contact with cells are called gap junctions. So we went from super tight junction to pretty tight, let some things through. And then now we have gap junctions that cells are touching, but it's this junction really isn't keeping them together. It's not so super sticky and strong. Really a gap junction, really a little adhesion. And that's pushing it. Mostly for communication. Okay. And as you can see from this cartoon here, here's cell 1, here's cell 2, here's these molecules. So the gap junction is actually composed of these proteins called connexins. Okay. And 6 connexins stand in a circle, hold hands, interact with one another to form a tube, a big giant tube. And that tube is called a connexon. Okay? So they just, 6 connexins interact with one another to form a tube called a connexon. And so this tube has the ability to allow molecules to travel, small ions, small molecules to travel from one cell to the next. Okay. And that's essentially your gap junction. So you have your 6 connexins, make a connexon. Here's what it looks like. And this actually is nice because it shows you the surface of the cell. So this is the cell, this is the inside of the cell here. You can see that the surface of the cell has many, it's just like a blanket of these. And here's just like one slice if you just cut the cells in half and we can see what was happening. So the molecules can go from here into the cell. Okay. I think that covers gap junction. So where would we find these? Oh, why would these be important? So where do you find them? Obviously we find them in the cell intestine because that's what we've been showing. So one example is in the intestine. Okay. But a really cool example that I love are your heart cells. So the heart cells are one next to the other. And the reason why we have this rhythmic heartbeat is because one cell just received the molecules from the neighbor cell and now is sending it to the next cell. And so every time the next cell receives that molecule, it knows to hiccup. and then it sends the molecules to the next cell, and that cell jumps up and down. And that's why you have a heartbeat, this rhythmic heartbeat. Okay? So cells that need to be in synchrony or that have rhythms. Okay, cool. So let's put it all together, well, at least what we've talked about today. So here are your intestinal cells. Again, this is like a slice if you were able to like cut through a cell. And if we just zoom in, you can see that, well, let's not zoom in quite yet, but you have 3 cells here. Okay, let's zoom in a little bit. And you can see that one cell absolutely has the potential to have all 3 junctions, and this happens all the time. So you do have your tight junctions, keeping for this intestinal cells, it's important to keep the top near the villi, needs to be super tight and not let anything through, because this might be the bloodstream, and then this is going to be the lumen, the empty space where like stuff goes through your intestine, right? So you don't want stuff from here leaving into your bloodstream. So you really have to have the surface of it really tight. And then you have your adherens junctions, right? So, or sorry, here, your adherens junctions, your desmosomes, and some stuff can get through. And then finally, you have your gap junctions, these tiny little guys down here. This is the nucleus, here's your F-actin, and this is the basement. So this is how cells, next on Tuesday, you'll see that cells also have to make sure that they anchor to that basement membrane. We'll talk about that. So it's important because if you don't have a really tight junction up here, boom, 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 you have stuff that you don't want entering places you don't want. So something called leaky gut syndrome, that's pretty uncomfortable if you do experience it. And why is it uncomfortable? Because it's food that hasn't been digested, or bacteria, or toxins, poison. It's poison for your gut. Okay, so we'll just stop there and we'll continue on Thursday. Any questions? Can different types of cells have adherens junctions between them? Yeah, but it's not as common. So adhesion junctions, so that includes the tight junctions, the gap junctions, the adherens junctions. Most of those are usually going to be between cells of the same type, so that we can, because usually cells of the same type need to stay together so they can function as a whole tissue. So I would say this is usually going to be cells of the same type, same type, same type. Not to say it's an exclusive list, but yeah, for the most part, same type. Okay. Any other questions? Okay. Well, let's see. What do we have on Tuesday? Okay, what'd I say? Okay. So on Tuesday, we'll talk about direct cell contact, but not cell to cell, cell to its wall, cell to its environment. Okay. And then we will move into how cells communicate with one another. Okay, cool. Yeah, all right. So that will probably take up the majority of Tuesday's lecture. So just keep that in mind. And for the DQ, I do encourage you to at least skim through the fall URL for lecture 9 so that you can get a pretty good idea of what's happening, so you can show up to class on Tuesday nice and prepared. Because this lecture probably does take a little bit more repetition so that you can fully understand. So at least 2 or 3 times of watching this. Okay, great. So tomorrow's a holiday, so we don't have class anyways on Fridays, but enjoy your 4-day weekend. So take care of business, but find some time for yourself and to have some fun safely with a mask on and social distancing, please. And I will see everybody. If you're my Thursday lab, we'll see you soon. And if not, then I will see you next Tuesday.